everyone, thank you for being here today. My name is Dylan and this is my partner Ilona. We will be presenting on circadian rhythms and the sleep wake cycle. This is a really fascinating topic because it connects neuroscience, biology, health, and even society. We interact with circadian rhythms every day, even if we don't realize it or not. Circadian rhythms are natural internal cycles that repeat approximately every 24 hours. They're involved in many body processes like hormone release, digestion, body temperature, and especially the sleep wake cycle. The sleep wake cycle is probably the most obvious circadian rhythm we experience. It's basically the pattern of feeling awake during the day and sleepy at night. What's amazing is that this whole process is regulated by an internal biological clock, but that clock isn't isolated. It's influenced by things like light exposure, genes, and even behavior. So, today we're going to walk you through the brain structures, genes, and hormones that control circadian rhythms. And then we're going to talk about two recent scientific articles, followed by societal impacts, ethical issues, and then we're also going to discuss what we still might not know.
So the brain structures involved is going to include these three main structures. The first one is the suprachiasmatic nucleus, which is the master clock and located in the hypothalamus of the brain. And the second one is the retinohypothalamic tract, which puts light input into the SCN, as well as the pineal gland, which produces melatonin in response to dark cues.
Circadian rhythms are not just controlled by the brain, they're also regulated at the cellular and genetic level. So, the four key genes that are involved are the clock, the BMAL1, the PER, and the CRY genes. Here's how they all work. So, the clock and the BMAL1 proteins bind together and they activate the PER and the CRY genes. As the day goes on, the PER and the cry proteins build up in the cell, and when they reach a certain level, they shut down the clock and the BMAL1 activity. Eventually, the PER and the cry break down, allowing clock and BMAL1 to start the cycle again. This whole feedback loop takes about 24 hours, and that's what gives circadian rhythms their timing. This genetic feedback loop happens in nearly every cell in the body, but it's coordinated by the SCN, which helps keep everything running on the same schedule.
So there are four main hormones and neurotransmitters that are involved in the sleep-wake cycle. The first one is melatonin, which is released from the pineal gland, and it is released in response to dark cues at nighttime, and melatonin helps promote sleep. Cortisol peaks in the morning in response to light cues, and it helps with alertness. Adenosine builds with wakefulness, and it helps increase sleep pressure. And light regulation helps inhibit melatonin production through SCN signaling.
The first primary source we talked about was titled A Mathematical Model Towards Understanding the Mechanism of Neuronal Regulation of Wake, Non-REM, and REM States. The researchers wanted to understand how sleep stages are controlled by the brain, specifically how transition between being awake, non-REM sleep, and REM sleep, and they used mathematical modeling of neurons to simulate the behavior of real brain circuits. What they found was that a certain neuron in the substantia nigra and oxyron producing neurons are very important for maintaining and switching between sleep stages. Their model accurately replicated the realistic sleep wake cycles. This is really important because it gives scientists a tool to predict how the brain might react to disruptions in sleep, like from stress, medication, or neurological disorders.
Our second primary research article is titled Neuropeptide Dependent Spike Time Precision and Plasticity in Circadian Output Neurons. And the researchers for this article wanted to research how neuropeptides DH31 and DH44 regulate circadian output. They did this by analyzing fruit fly brains as well as optogenetic stimulation. And they found that these two neuropeptides, DH31 and DH44, help increase precision in PI neurons, as well as improve accuracy in the encoding sleep-wake signals through the SCN. This is important because it highlights how chemical signaling can control clock neuron output as well as sleep regulation.
We also looked at three secondary sources. The textbook Neuroscience exploring the brain helped us understand how brain structures like the SCN function. The NIH explained how the clock gene feedback loop works at a molecular level, and the National Sleep Foundation gave insight into how things like light exposure and bedtime habits affect our rhythms. All of these secondary sources really helped us connect the research to real world applications and things that we see in our everyday lives.
Some disruptions to the circadian rhythm can include things like night shifts, say you're working the graveyard shift at your job, or jet lag, as well as excessive screen time. And some health effects that can come from these disruptions can include insomnia, depression, metabolic issues, as well as weakened immunity. And a fun little statistic is that 70 million Americans suffer from sleep disorders.
So, these disruptions don't just affect individuals, they affect society too. As an example of this, drowsy driving, poor work performance, and lower academic achievement have all been linked to poor sleep. There is also an economic cost. A RANAD report estimated that sleep related problems cost the US about $411 billion a year in lost productivity and healthcare. Some solutions include blue light filters, light therapy lamps, and melatonin supplements, but these also are not cures. They can help, but we do need a better understanding of the biology behind these rhythms in order to create potential cures.
So, there are a few ethical implications that come from altering the sleep cycle. One of them is that 24/7 work cultures can be harmful to workers' health. And is it ethical to implement a 24/7 work schedule at your job? And should we be medically manipulating circadian rhythms through methods such as pills or implants? And if we do decide to medically manipulate circadian rhythms, who would have access to these treatments and technologies?
Even though we've learned a lot from our research, there's still gaps in current research. One big question is why different people have different circadian preferences, like being a night owl versus an early bird. Is it genetic, environmental, maybe a combination of both? Another issue is that most research is done on animals or small specific populations, and we need more diverse and human focused research to get some of the answers that we're looking for. Also, new methods like optogenics and fMRI can help us understand the SCN and other clock systems in more detail.
So, in conclusion, our hypothesis was that sleep-wake cycles and circadian rhythm are controlled by the brain's response to light cues in the environment as well as melatonin levels. And some supporting evidence for our hypothesis is that the SCN located in the brain will act as the master clock, and it will receive direct input about light cues through the RHT. The SC then will regulate the pineal gland's production of melatonin, which promotes sleep during darkness, and the molecular feedback loops involved in the genes that we mentioned previously will help support the timing of these rhythms. And then hormones such as melatonin and cortisol will fluctuate as the light and dark cycles to help regulate that feeling of alertness and sleepness.
Here are our references. Again, we used 2 primary research articles and then 3 secondary articles. And we thank you for listening to our presentation. Hope you enjoyed.